So while they're setting that, I'm going to make some very brief introduction about research in general in the scleroderma field. Then I'll introduce Dr. Maureen Mayes, who's a professor of medicine in Houston and has been one of the leading scleroderma researchers and really one of the pioneers in using genetic approaches to understand scleroderma, and as well as leading clinical trials. So she'll talk about that. And then Dr. Robert Lafyatis, a professor of medicine at Boston University, who's also a pioneer in translational research, and he will bring us up to date on some newer clinical trials. And then I will end up by talking a little bit about some of the emerging new areas of research currently. But I think it's fair to say that we've made a lot of progress in scleroderma, and that's shown in this slide, which is that if you just look at the 5-year survival in scleroderma, just over the past decade or so, there's been a very significant improvement. So clearly people with scleroderma are living longer and better and more functional, and that's the good news. But the challenge is that still we remain without an effective disease-modifying treatment, and as I'm sure you're aware, there's no single drug that's been approved by the FDA for the treatment of scleroderma. And this is illustrated in a clinical study, and I'm not going to bore you with the details, but this is a study to look at treatment of lung disease in scleroderma. This was considered to be a positive trial because the patients who got cyclophosphamide clearly did better than the patients who got placebo. So that was exciting. But if you look at the actual difference between the treated and the untreated group, it's very, very small. And this highlights of how many of even our relatively effective drugs are far less than magic bullets. And in fact, if you look at many studies, even Cytoxan, this drug that was used in this study, shows efficacy perhaps in stabilizing the lung disease, but very rarely really improving it. So we have to do better. And so the major challenges in scleroderma today are obviously to discover and implement effective disease-modifying treatments, and that's intimately tied to the second question, which is that we need a better understanding of what this disease is, what the molecules are, what the pathways, what the cells, what the genes are. And a lot of the current research really aims to provide a better understanding of that. Now, the next two talks will really focus on development, discovery, and validation of new treatments. And this slide just shows a highlight in a very broad term of how this kind of drug discovery research is done, and showing that there's an initial preclinical phase that's done in the laboratory. And here, typically, we use test tubes and explanted fibroblasts and experimental mice, very basic scientific research. And if those look promising, researchers then move to the next phase, which is often called translational research, which is really focusing on trying to understand how these findings may be applicable to a disease like scleroderma. Now, if those results look good, and this can take months to years of work, often a promising target or drug will then move into early clinical trials that you'll hear about in more greater detail. And ultimately, the few drugs that look good and show positive results in these clinical trials move to the bottom, where they become accepted and approved for indication and become widely used and hopefully affect the outcome of this disease. So you'll hear more about these pathways, and with that I'll turn it over to Dr. Maureen Mayes, who will talk about the basic ideas of clinical trial design and how these clinical trials are done.
Well, thank you, and it's a pleasure to be here. We've got my slides up. We're going to talk about the overview of scleroderma clinical trials, and I have been involved in this. I was just trying to figure out how long it's been. I've been working in scleroderma in terms of treatment for the last 30 years, and I think about 20 years ago we published the results of our one of the first randomized controlled trials. It was multi-center in scleroderma. We'll talk about that one in a few minutes, but it's been a long and difficult road. The basic elements of a clinical trial, Dr. Varga just mentioned the preclinical phase, and this is in the test tube and in looking at mice to see if experimental forms of scleroderma can be helpful or if a drug is active in the test tube. That's the preclinical phase. And then the phase 1 clinical trial involves a small group of individuals. And really, this is the first time in humans trial, so this is really safety. And after that, you go to a phase 2, a larger group, and at that point, you're asking not only is this drug safe, but is it effective in the disease that you want to study? And phase 3 is sort of the classical blind, randomized, controlled trial that will definitively answer if a drug is both safe and effective in the disease that you want to study. And then there's post-marketing surveillance, and I think this is what tends to hit the news every once in a while. There'll be a drug that's withdrawn from the market after it was approved, because only when it was tried in hundreds of patients or perhaps thousands of people that suddenly a side effect became apparent that wasn't apparent even going through phase 1, 2, and 3 trials. But it was only after it was on the market for a period of time that people realized that it caused some other problems. Is there anybody in the audience who's been part of a clinical trial? Can I see any hands? Ah, yes, good. And I want to thank you for helping, because you are really helping research in a way that none of us here can. Well, the gold standard is the randomized, double-blind, controlled trial. And what that means, to be randomized, each person is assigned to a treatment arm by chance, like the flip of a coin. And these days, that's done by a computer. If we have a patient who meets the eligibility criteria and they're willing, they've signed the consent form, then we put their information into the computer. Usually this is a multi-center trial, and then we get back a number that tells us which of the 2 arms or 3 arms that this patient is going to be assigned to. Or actually what it does, it gives us, if it's a pill form, then we're told that this is patient number whatever, and we go into our cabinet and take out that number to give the medication to the patient. We'll talk a little bit about what blinding means, but randomized, it's neither the patient nor the doctor can determine which treatment the patient will be on. And what the purpose of this, it avoids what we call a bias, a randomization bias. If it is left up to me to determine which arm a patient should go into, there will be, if not a conscious, then at least an unconscious bias, that perhaps I perceive this individual as being sicker, and I don't want them to be on a really aggressive medicine, or maybe I do, but I would, if I had to make the choice, then I would choose one over the other in a way that wasn't totally random. And if the decision was left up to the patient, then they might perceive themselves as being willing to undergo certain risks but not others, and they might choose one arm rather than the other. And when it means, when we say double blind, we mean that neither the patient nor the doctor knows what arm of the study the patient is in. And the pills, if you're looking at drug A versus drug B, even the pills themselves are made to look alike, so that the patient or the doctor can't look at the bottle and say, oh, these are blue pills rather than green pills, so the patient is likely to be on one versus the other. And it's hard to blind a study if you're comparing an IV medicine to a pill medicine or a procedure to medication. And sometimes it's impossible to blind it, because if the study compares a surgical procedure to pills or to IV infusions, then there's no, you know what arm the patient is on. And this is true. We were part of the Scott trial, which is the scleroderma cyclophosphamide or transplant trial. And people were randomized. One group got high-dose chemotherapy followed by stem cell rescue, and the other group got IV chemotherapy once a month for 12 months. Well, obviously, it was impossible to blind that. Sometimes the study design will try to do that because they'll say that the treating physician, of course, will know what the patient is on, but the person who is determining if it is effective or not, say if we're looking at skin scores in scleroderma, then that person coming in to do the skin scores won't know what the patient has had in the past, and that also means that that physician doing the skin scores can't talk to the patient because the patient might say, oh, well, when I was at, getting my IV infusion last month, oops, suddenly they become unblinded. So for the Scott trial, we didn't try to blind things. We knew it was impossible, so we just went forward knowing that we could randomize people to one of the two arms and that would be that would be good enough. Now, to have it be a controlled trial means that you're comparing one agent to another. The most common one that you probably have heard about and you might think about in a clinical trial is a placebo-controlled trial. So one group gets what we hope is active medication, and the other group gets a dummy pill. The other way to do that to a controlled trial is to look at a new drug versus standard of therapy. We did the scleroderma lung study that Dr. Varga just alluded to a few minutes ago, and in that was a placebo-controlled trial. So people either got the cyclophosphamide pills or they got a pill that looked exactly the same, but it was placebo, it was a dummy pill. And that showed that there was some effectiveness for the Cytoxan. We're now doing a follow-up to that study, scleroderma lung study 2, when we're comparing the Cytoxan or cyclophosphamide to a medication called Cellcept, which is also known as mycophenolate, because there's some evidence that we know that the Cytoxan is somewhat effective, although there are potential side effects. It looks from some studies that mycophenolate or Cellcept is also effective, so we're doing a direct head-to-head comparison, one to the other. So although it's a controlled trial and it's randomized and blinded, still there's no placebo arm. You either get one true medication or the other true medication. And finally, you can use combination therapy versus a single drug. And one of the reasons that we do controlled trials is because of the placebo effect. In any trial, even in scleroderma trials, there are some people who are going to be stable. Even if you didn't give them any medication, they're going to be stable, so that if you look at their skin scores now versus the skin scores a year from now, they're not going to change much. If you look at pulmonary function tests now versus a year from now, that's not going to change that much either. So you need to have the same group of people with the same likelihood of getting better or worse or staying the same, and then you compare one either true medication to placebo or one medication to the other and see if one group gets better or not. Our trials now also have questionnaires that ask patients how they perceive themselves to be. Do you feel that you're better or worse? Do you feel that your breathing is better now or stable or worse than it was at the beginning of the trial? And when they've done trials in any disease, in scleroderma and rheumatoid arthritis, doesn't matter what, there are people who feel better after they've been on medication or they've been in a study, but that their subjective experience makes them feel better, but in fact, they were on placebo all along. So it's really important to have a controlled trial and a controlled arm. Frequently, studies are begun because one medication looks like it should be effective in a disease like scleroderma, or because the mechanism of action is such that we figure, well, it ought to be effective. So initially you try it out in a group of people in a non-randomized, non-blinded, non-controlled fashion. And you look at, and you can see that perhaps more people get better than you would expect by chance alone. That's a single open label trial, and some of our controlled trials are based on evidence from these case series of open label treatments. But over and over and over again, when we've tested those medications in a controlled setting, we find that they are no better than placebo or no better than standard care. We'll go over that in a minute, but it really brings home the need to have a control arm in order to compare the results of your new medication. And then there are ethical studies, or ethical issues. All studies have to be approved by the Institutional Review Board, this Human Subjects Committee, at all the institutions of the people, the investigators. All patients have to sign a consent form. When I was at Wayne State, just starting out doing clinical treatment trials, the Human Subjects Committee insisted that all consent forms be 1 page. That is ancient history now. The consent form for the Scott trial was 21 pages. And I think that is really far more than any individual can really understand. We give people the consent forms to take home, to go over, to discuss with their family members, to get questions and to come back, and we can address those questions. But the consent issues are really very serious. The baseline, or the bottom line for any consent is the following. You have to be told, 1, this is a study, and it is research. You don't have to be in the study. The consent form has to outline the risks that are associated with the study, as well as the potential benefits. And you have to understand, you don't have to participate in the study. That if you decline to participate, that it's okay, that I'll still be your doctor, that you can still come here, that there are no negative consequences for you saying, no, I really don't want to be in the study, I don't want to take these risks. So let's talk about some scleroderma clinical trials. And this is the evidence for randomized, blinded, placebo-controlled, or other controlled, multi-center trials, and we're looking at fibrosis, not vascular disease, not pulmonary hypertension, but fibrosis in scleroderma, either in the skin or lungs. The first trial was the D-penicillamine trial that now was reported 21 years ago, was the first multi-center trial I got involved in. There was a photopheresis trial that was also controlled. There was an oral methotrexate trial, and methotrexate's a common medication for rheumatoid arthritis. There was an interferon trial, a relaxin trial, there was a collagen trial, something called anti-TGF-beta. There was BILD2, which was bosentan in interstitial lung disease, and then there was SLS1, the cyclophosphamide trial. And this goes now from 1991 through about 2006. And what were the results? Well, unfortunately, the D-penicillamine trial was negative. The European trial was also negative for pheresis. The methotrexate trial was negative for fibrosis. People with scleroderma can get benefit from methotrexate if they have inflammation, especially joint inflammation. So you can be on methotrexate, you can feel better, but it's not going to influence the skin or the lung fibrosis. Let's see, interferon, relaxin, all negative. Now, this is 20 years of my life of treating trials, and every time we started a trial, there would be this, this should really work. We have all of this evidence. It helps mice in the test tube. It improves scar tissue formation. Why can't it? So we're going to do it in a controlled setting in humans and see what happens. Okay, the TGF-beta trial was negative. The BILD2 trial. Now, you can understand that we got very excited when we finally broke the blind and looked at the results, analyzed the results of the scleroderma lung study 1, the cyclophosphamide trial, because that was the first time we were able to demonstrate, even though the effect was modest, that we were able to demonstrate that there was any effect in scleroderma looking at lung involvement and also looking at an improvement in skin disease. So the good news is that we had a positive trial. The bad news is that it was cyclophosphamide, which was originally developed and is still used as a chemotherapy for cancer and has a lot of potential side effects associated with it. I'm going to mention a few ongoing trials for scleroderma fibrosis. There was a Scott trial, the scleroderma cyclophosphamide or transplant trial, which is no longer recruiting patients. The last patient was recruited now a little over a year ago, but the outcome measure is determined at a little over 4 years. So we have to follow all patients recruited for 4 years before we can analyze the data and be able to say that that one arm of the trial is effective or the other. And as I mentioned, there's the scleroderma lung study 2, SLS2, that's comparing 2 active arms. And there are some others that I'm not going to talk about now, but there are other trials that are ongoing that have some potentially promising drugs being used, but we won't know until we have recruited patients and then done the analysis to see if these agents are in fact effective. But let's look at one other aspect of scleroderma, sort of the other 50% of problems that scleroderma patients have, and that is the circulation problems. The Raynaud's, digital ulcers, pulmonary hypertension, and here the results have been much more promising and much more positive than looking at medications to influence fibrosis. There are multiple medications in good controlled clinical trials that have been demonstrated to be helpful. Calcium channel blockers, this is a classification of medications that most people with scleroderma are on and is effective in helping the Raynaud's. None of these medicines can cure Raynaud's, but they can be helpful to decrease the attacks and to improve healing of ulcers or to decrease the number of ulcers that develop. And then for pulmonary hypertension, bosentan, and others. So this is for Raynaud's, and the real success story has been in pulmonary hypertension because multiple studies have shown that there's been improvement in pulmonary hypertension. There are now 3 classes of medicines that are out there, all of which have been shown to be effective. They use 3 different mechanisms of action, and there are now multiple drugs in each of these categories. I won't go over them, but at least we have multiple medications to choose from when we diagnose early pulmonary hypertension in our patients. Future trials that will be talked about by our next speaker will also, the number of trials in scleroderma that are actively being conducted has increased a great deal since I started in scleroderma now about 30 years ago. We are able to organize scleroderma clinics and do multi-center trials to recruit patients, to follow patients carefully through the studies, and to generate really good data that we can then determine if these medications are helpful or not. So, in summary, when you look at circulation, especially a problem like pulmonary arterial hypertension or PAH, we've really been very successful, or pharma has been very successful in developing these drugs, and the research community in scleroderma has been able to demonstrate that they're effective. But studies of drugs for the fibrosing component of scleroderma, the pulmonary fibrosis, the scar tissue in the lungs, the skin scar, fibrosis, those studies have been less successful. But the research continues. There's more clinical trials now than ever before, certainly in my professional lifetime. And I think that I am optimistic that in the future we're going to have more trials and better trials. And I think that's it. Thank you very much.
So I'm going to talk a bit about ongoing clinical trials and a little bit actually about trials that are going to be planned in the near future. And I apologize a little bit. Some of this will seem a bit list-like, and I think initially what I'll do is try to provide you a little bit of background similar to what Maureen spoke about, but some of these things are worth going over more than once, I think. And so we think about trials or I think about trials anyway, as sort of two general types: early phase clinical trials, which are designed primarily to see if a medication is safe. And what we'd like to do in those trials is try to understand a little bit about whether there's hope to bring them into larger trials, because larger trials are basically expensive and we can't study every drug in a larger trial. These early phase trials are oftentimes the first time the medication has been tried in scleroderma. Generally, they're small, 10 to 20 patients. They may not be placebo-controlled, so if you come into that sort of trial, you may know ahead of time that you're going to get the medication, in contrast to the later phase trials. And they're often single-center, so the disadvantage for you if you're interested in this sort of trial is that it may not be going on at a center near you. Late phase trials, which are typically phase 2 or phase 3 trials, and phase 3 are kind of gradually getting bigger, if you will, and these phase 3 trials are the ones that typically lead to FDA approval, so they're very important and very carefully monitored and have to live up to a higher standard in terms of whether a medication is actually helping. And those, as Maureen had mentioned, are randomized and placebo-controlled trials. These are generally multi-center because in order to recruit enough patients, you have to have more than one center involved. And more likely, you're going to be having a center close to you that might have that kind of trial going on. Trials are going to also typically target different parts of the disease. So your problem might be your skin, your problem might be your lungs, or it might be a number of different manifestations. The manifestations of scleroderma that we've been studying most have been skin disease, interstitial lung disease, pulmonary arterial hypertension, digital ulcers, and to some extent Raynaud's. There are lots of other complications of scleroderma that haven't been looked at carefully. Gastrointestinal disease has, in most cases, not been looked at carefully. Fortunately, some of those manifestations we can control relatively easily with acid blockers, but unfortunately, some of them have just lagged in our ability to study them. So this is just a limitation to what we can focus on. My hope is that if we can understand maybe skin disease or find a good antifibrotic agent for skin disease, or for that matter for lung disease, that might treat the underlying process and help other manifestations as well. So if you're interested in a clinical trial, and certainly I think all of us here would agree that these are fundamentally extremely important for our advancing therapy for scleroderma, there's a variety of resources you can go to, and I'm going to, after this slide, I'm going to start looking at various trials, but you have a number of resources which are probably as good or better than these slides, and those are your scleroderma rheumatologist, and hopefully, I think most of you, if you're at this meeting, probably have a rheumatologist who specializes in scleroderma that you've seen. Those doctors often are going to be aware of the kind of clinical trials that are ongoing in your city or in your region. The Scleroderma Foundation is an excellent source also for information about clinical trials. And I think the motivation for getting in clinical trials is a variety of different motivations, but one is that you may be looking to try something that might help you that you haven't been able to find so far. I looked actually myself at, I put nih.gov, but what it is clinicaltrials.gov is the website, all one word, clinical trials. And that website categorizes all clinical trials going on in the United States as well as many international trials. And if you put in scleroderma into the search engine for that site, all trials, and there's about 161 trials listed right now, many of which are completed, but we'll go through most of the active trials listed in that site here in a few moments. And I'm going to put a little plug in for our website. Boston University now has a website, the CORT website, CORT, that also has a listing of at least the clinical trials going on in the New England area. So if you're outside of our area, not so useful. So now, I apologize, this is going to be a bit list-like. You're going to have a chance to ask us questions later, but I'm going to try to speak mainly about some of the ongoing major clinical trials. And there aren't so many, but I think, as Maureen was alluding to, and I think one of the really exciting things about working in scleroderma research at the moment is that it's becoming a real focus for not just the government and the NIH, but also for companies. Companies are interested in scleroderma now, and a lot of medications are being made available, or money being made available to try them in scleroderma population, in patients. So one of the trials that's ongoing, and this is funded actually by the NHLBI, one of the branches of the National Institutes of Health, the National Heart, Lung, and Blood Institute, is funding the study comparing cyclophosphamide and mycophenolate, called SLS2. These are now studies looking at interstitial lung disease. Looks like I put lung down there twice. Sorry. The second major study that's just being started now is pomalidomide. So this is actually a derivative of thalidomide, which is a drug that Celgene has made for a number of years. As you remember, this was very unfortunately a drug that led to fetal malformations, as was taken off the market years ago. But pomalidomide is a much more potent relative of thalidomide that they think might have a good effect. And some doctors have used thalidomide in scleroderma. It's never been subject to a controlled trial, so that there's never been information really clear about how good thalidomide would be. This medicine hopefully will help answer that question, as well as whether this drug really will be good for lung disease. This is a multi-center trial, so it's likely that if you have interstitial lung disease, that you might be able to fit the criteria for this trial, and particularly if you have progressive lung disease, might be able to enter that sort of trial. Many sites across the country are going to be participating in that trial, as well as the scleroderma lung study. So there's a variety of other much smaller trials that are targeting interstitial lung disease, and I think maybe not as much worth trying to go into any particular detail. There's a small open-label trial at University of Michigan looking at a medication called N-acetylcysteine. There's a couple of different institutions that have been taking different tacks on trying to understand better whether stem cell transplant will be useful. And there's been an individual at Northwestern for years that's been studying different ways of treating prior to stem cell support, and there's an ongoing single-site trial for that. Both Duke and University of Washington, who've been leaders in the Scott trial, are trying what's referred to as allogeneic stem cell transplant, and without trying to go into the details of that, it's a variation of a stem cell transplant. These are quite early and quite experimental, and I wouldn't, unless, I think for most patients, those aren't going to be things they would probably want to get involved in. So what's going on in targeting skin disease? And this is an area that I've been quite actively involved in. And first and probably foremost is this tocilizumab study. This is a study that Hoffman and Roche took upon themselves, started relatively recently, but they're recruiting for it as quickly as they can. And it's trying a medication that was recently approved for rheumatoid arthritis called tocilizumab. It targets a cytokine, and many of these new medications we know what the targets are. They actually block certain molecules that are involved in mediating normal inflammatory processes, and we think that they may be out of whack, if you will, in scleroderma and causing part of the problems. Anyway, it's clear that in patients with rheumatoid arthritis, if you block IL-6, that they get dramatically better. And the question is, if we block IL-6 in scleroderma, will patients get better? This is a multi-center trial. It's placebo-controlled. It's considered a phase 2 trial, so they're skipping the phase 1 in this case. They're not really so concerned about safety, which is what phase 1 trials are often about. This will probably be enrolling for another, I think, 6 months or so, maybe longer. And I think it's a great trial. It's going to be a very important trial. It is placebo-controlled trial, so half the patients who enter this will receive placebo, whereas half will get the active medication. my institution, I've been leading a series of small, early open label trials of medications that are quite early in development or thinking about for scleroderma. And the two we have actively going on right now is an anti-TGF beta trial, which again is an open label trial. It's small. We'll be recruiting for this probably not a lot longer time. We're only entering a total of 16 patients into this trial. Patients with skin disease with relatively recent onset scleroderma are eligible for this. And our goal is to, in part, to prove that this medication might be useful, but also in part to try to prove that this is an important target and that this TGF beta is actually important in the disease pathogenesis. We have another sort of, not really companion trial, but very similar structured trial using a drug on the market that's already approved for other indications, rilonacept, and testing this also. It blocks IL-1, interleukin-1, rather than interleukin-6. You'll recognize as you hear much about different drugs available right now, a lot of them block things called interleukins. These are chemical mediators that are responsible for stimulating inflammation. And so we're actually, I don't know what the highest interleukin right now is, but I think it's on the order of 36 or more. The first one discovered was interleukin-1, and it still looks like a reasonable target anyway in fibrotic disease. So we're testing that in a small open label trial as well. That's, again, a single-site trial at Boston University. There is, I mentioned a small trial, and there may be more of these going on that I was aware of at Feinberg, where they're looking at the effect of mycophenolate on gene expression in the skin, and that's, again, an open-label trial. If you talk to your local your scleroderma rheumatologist, they may be aware of other trials that I don't know about going on in skin disease that maybe aren't on NIH.gov. I do know there's a lot of very exciting and what I consider very exciting trials that are going to be started in the near future, most of which actually I'll be involved in, but I think everybody sitting at this desk will be involved in many of these, because most of these are going to, all of these are going to be multi-center trials. So abatacept or abatacept, depending on how you like to pronounce this, and I've not got clear really what the pronunciation is. This is also known as Orencia. It's another medicine that's been approved for rheumatoid arthritis, and you can see that sort of a pattern emerging there that once a medication's been shown to work in what we consider a related disease, then it kind of makes sense to try it and see if it works in systemic sclerosis, in part because the safety is known. It's known to have an important target that works at least in some other related disease, and so it makes sense to try it in scleroderma. Dr. Kahn at University of Michigan is going to be the principal investigator on this study, which I think is going to start up fairly soon. In fact, we're almost in a position right now where we have more trials than we know what to do with, and I think that's actually a good position to be in. I'm very, very excited about that position. We hope as we look at multiple agents in scleroderma, and I think this is really a paradigm shift from what you saw Maureen showing for, back in 1990 to 2006, a drug was tried. We hoped it would work. It didn't work. And then another drug was tried, and we hoped it would work, and then it didn't work. And then and so that sort of serial trial process was pretty lengthy and slow. And the answers to the questions were quite clear. I mean, the studies were done very well. The answer, unfortunately, was that it didn't work. And I think what we're seeing now, in part because of industry interest and as well as corporate interest, is that we see multiple agents that are coming forward. And part of the real challenge now is to do each of these studies carefully enough so that we don't miss the good drug. And it's not trivial sometimes to tell whether or not you're getting a good response. It may sound really easy. I feel bad or should be enough, but actually it's not necessarily always easy for us to tell when a medication is working in an early phase trial. Do we have enough patients in the trial if we conducted the trial properly? But in any event, we have abatacept, we have an LPA inhibitor by Sanofi with this rather bland name, SAR 1000 or 100842. It probably has another name, I'm not sure what that is. That's again going to be a multi-center placebo-controlled trial, I think going to start within a couple months. And then I've been speaking with Pfizer about an anti-IL-13 study that I hope will be starting relatively soon as well. So these are all, I think, exciting developments in looking at skin fibrosis. And although we think about drugs targeting skin or targeting lung, that's a bit artificial because we think and hope that drugs that work for the lung will also work for the skin and vice versa, because we think the mechanisms that underlie both of these processes are quite similar. So we, to some extent, arbitrarily choose to study one manifestation or another of the disease, but the likelihood is that they may have a broader use. So switching to pulmonary hypertension, ongoing trials for pulmonary hypertension, I was surprised that there actually weren't more of these because I think there have been a lot of pulmonary hypertension trials, although to be honest, a lot of the pulmonary hypertension trials that have gone on over the last few years have been really focused on those several agents that Dr. Mayes showed you have already really been approved for, or 3 classes of agents that have already been approved for pulmonary hypertension. So we know that PDE5 inhibitors and so anyway, those 3 classes of medicines work. Now, a lot of the trials have been sort of variations on those, and actually the second trial there I'm showing is another variation on that, ambrisentan and tadalafil. These are medicines we know already work for this drug, for pulmonary hypertension. The question is, does using them in combination work better? And that's a single-site trial, but probably the most exciting trial at the moment is the rituximab trial. This is a NIH-sponsored National Institute of Allergy and Infectious Disease. This is actually a different branch from what we normally think of as being our scleroderma branch, which is the NIAMS, which never seems to have enough money for us, we feel like. But these other institutes actually have bigger budgets, frankly, and the clinical trials are expensive, and they have an interest in scleroderma, and fortunately we've been able to tap into that, or different investigators have. Anyway, Dr. Metzger is the sort of the rheumatology PI on this study, and there's a couple other pulmonary, I think, who are pulmonologists who are leading that study. It's a multi-center study, I think a good study to get involved in if you have pulmonary hypertension to think about, and likely because it's multi-center, there might be a site close to you. I just mentioned there's another study going on at BU looking at, it's more of a biomarker study, and for the most part, I'm not speaking about biomarker studies. There's lots of those kind of studies going on as well. These are mainly, I'm speaking about here, are interventional trials that are designed to hopefully find a new drug that will work for you. And Maureen also mentioned this clinical trial. So clinical trial for digital ulcers, there's only one that I'm aware of. It looks like, as best I can make out, and maybe Maureen can help me with this, it looks like Dual 1 and Dual 2. I think these are basically the same trial. The company's decided it wants to get this to FDA approval quickly. And so what they're doing is 2 multi-center trials that look very similar, and they're big enough that they're hoping to get FDA approval quickly using this drug macitentan, which is a relative of ambrisentan, of bosentan. It's one of these what are called endothelin receptor blockers. And they're trying it in 2 different doses versus placebo. So in this case, the placebo arm, you'll only get put into that 1 chance out of 3, which is, I think, a nice trial design. Most of us, I think, feel, at least my sense is most of us feel lucky and are going to get put in one of the, think it's lucky. Should mention one of the concepts behind all the clinical trials we do is this concept of equipoise, which means that in theory, the only time you're going to be put in a trial in which there's a placebo control is a setting where we really don't know that the medication has any benefit. So although most of us would prefer to be in the active treatment arm, we think that until we really know the medicine's helpful, that it's not unreasonable to have although if you get macitentan, you're in sort of the active treatment arm, there's also the risk of side effects from the medicine. So we think of those as kind of equally good places to be, put it that way. Only reason I came across this study was because I looked through clinicaltrials.gov, and if you're interested in clinical trials, I urge you to go there. You can put in not only systemic sclerosis as your search feature, but you can also put in your individual problem if you like. You can put in, for example, interstitial lung disease, and then just see those trials, and only those handful of trials that are looking at those questions will come up. But in any event, looking at a more extensive review, there is a trial going on at UCA Children's Hospital for children with systemic sclerosis that I frankly wasn't aware of, a CAMPATH trial for refractory systemic sclerosis, a very strong immune suppressive agent, a pilot trial. That's the only trial I think going on in interventional trial going on in childhood systemic sclerosis, which of course is extremely hard to study because there fortunately aren't very many children with this disease. So, in conclusion, there's really quite a lot going on, and scleroderma, as I mentioned already, is a focus for drug development by a number of different quarters now, not the least of which is drug companies who are literally wearing me out with their queries. I mean, there's just an enormous amount of interest by pharmaceutical companies right now in studying the disease, and that's good news for you, for all of us, really. We hope that some new trial designs will allow us to more quickly figure out which medicines are the right ones to put our money on, and so the preclinical trials that direct us hopefully will enable us to pick the right drugs to study in more detail. Our target is really to cure the disease, and we feel, all of us here feel pretty passionately about that. We unfortunately don't understand the disease well, but we do have just a tremendous number of new medications coming on the market, each which target different pathways, and which therefore potentially might work in the disease. We have never targeted that pathway previously. And the paradigm I see here anyway is multiple parallel studies. I don't think the series, as we've had to experience in the past, is really the way to go anymore. But to have many studies going forward at the same time as already occurring. But I see that, if anything, expanding in the future, so that hopefully we'll then narrow down what's really the single agent that's going to make the difference. So, thank you.
I'm going to take this back a little bit. So we talked a lot about drug trials and how we validate drugs and see if they're ready for clinical efficacy. And now I'm going to take it a little bit further back to the research, because I think it's evident, though, that we still don't have, we have some promising candidates, but we still don't have very effective drugs. And I just want to introduce this concept that currently we think there are two real distinct approaches to doing this kind of biomedical research. And simplistically, you could call one the bottom-up approach and the other one the top-down. In the bottom-up approach, you start with a hypothesis and you study cells, mice, experimental systems in the laboratory. You ask a question, you test your hypothesis, and test whether in this system your hypothesis is true or not. And if it is true, then you can go to the next step and validate that in other systems. The other type of research is the opposite, is what's called the top-down approach. And the interesting thing here is that it's a so-called hypothesis-free approach. You don't have any preconceived idea. You don't know what the question will be. And you simply collect as much data as you possibly can by looking at DNA, blood, serum, tissues, and measure everything you possibly can with the hope that something interesting will shake out. And if it does, then you can then formulate a hypothesis. But you can see that these two complementary pathways are actually very much interrelated, because in the top-down approach, you're going to generate a hypothesis, then you have to test the hypothesis in the laboratory. You have to examine how it works, what that molecule does. And vice versa, when you discover something in the laboratory, you have to take it back into populations and see how it really plays out in patients with scleroderma. And we see the interplay of these two approaches if you look at the history of scleroderma research. So it started out over 100 years ago with astute physicians and pathologists just looking carefully at patients and looking at tissues from patients. Sir William Osler was one of the great physicians who made many of these early observations. And it wasn't until really the 1960s and 1970s that physicians started to develop models that could be used in the laboratory, such as getting tissue from people with scleroderma to start testing hypotheses and discovering antibodies. That led to the development of mouse models. We don't have perfect models, but at least some mice recapitulate some of the features of scleroderma until we get to the current era. And the current era is really incredibly exciting, because what we see is the application of very sophisticated technologies that are called omics, and I'll come back to that a little bit, that allows us to measure millions of pieces of DNA and protein and different molecules to try to understand what they are actually telling us about the disease. And this was all made really possible by microchip technology and the development of computational approaches. And so what you see in scleroderma, like in other diseases, is an evolution in research from a very observational, empirical research to a very hypothesis testing kind of research that's called reductionist biology, to where we currently are what's called the systems biology that really attempts to integrate all of these pieces of information into one powerful predictive model. Now, just very quickly, the central dogma of how health and disease occur is very simply illustrated here to show that we all have the DNA blueprint that determines who we are. But then the DNA gets translated into RNA, which is the message, and it's the message then that regulates the proteins that really execute the biological functions of our cells and tissues in health and disease. And because of these powerful technologies, we now have ways to measure all of the DNA genetic variability. That's a field called genomics. We can measure all of the RNA, all the 30,000 genes that are being expressed, and that's called transcriptomics. And we can begin to measure many or most of the proteins that are present in the blood, for example, in a field called proteomics. So you can generate a lot of data, but it's not just an intellectual exercise for its own beauty. The point is to develop what's called systems biology that really integrates all of these kind of data with the hope that a predictive model can be developed that then allows us to tell which individual is likely to develop scleroderma, which patient is likely to develop pulmonary hypertension, which patient is likely to respond to a certain drug or not. Just to very quickly run through the 3 levels of this omics that we talked about. The first omics is looking at all the DNA, and there are 2 popular current approaches. One is called genome-wide association studies. The other one is the candidate gene approach. The genome-wide association study is really remarkable. It allows you to look at millions of pieces of tiny variations in the DNA and essentially see which one, if any, identify or associate with people who have scleroderma. And Dr. Maureen Mayes was a pioneer and led one of the largest efforts to date to look at all the genes in the human genome. This is a huge study that involved something like 3,000 patients with scleroderma and 5,000 healthy controls, each of whom had DNA, and the DNA was then analyzed. And what this complicated graph basically shows is showing across all the chromosomes which are the genes or locations in the genome that seem to be associated with scleroderma. And you can't really see this, and it's very difficult to see, but essentially analysis showed that there seem to be 5 genes or genetic locations that are more commonly seen in people with scleroderma than in healthy individuals. This is very complex and noisy kind of observation, so it's critical that these studies are validated, and many of these validation studies are currently ongoing. But the bottom line is that many of these genes have been replicated in other studies. So we know that they are real. So what do these genomics tell us? They will probably not give us predictive medicine. We will probably not be able to look at your genome and accurately predict whether or not you will get scleroderma using these approaches. However, they do tell us very powerful biological insights into what this disease is by identifying these genes and identifying genes that we never thought might be associated with scleroderma really triggers and stimulates further research into understanding what these genes do and what their function is. This is a very active area of research, and a lot of things are going on. There are additional genome-wide association studies, very large, expensive studies that are ongoing. And now we're at a stage where we can actually sequence the entire genome for close to $1,000. Used to be well over $100 million to do one genome. Now we're down to about $1,000. And those studies are now beginning, so we'll be able to look at every single genetic locus in the genome to see which are unique or different in people with scleroderma compared to healthy individuals. Now, the next level that is in this biological pathway is transcriptomics. Again, that's looking at all the RNAs that are being expressed and looking at whether they are different in healthy people versus people who have scleroderma. It sounds very, very complex, and it is, but in fact, technologically, we now have these chips, DNA chips that have all the genes present on them, and all you really have to do is get the tissue or the blood, put it on these chips, and you get a complex signal that really tells you the level of RNA in an individual with scleroderma compared to a healthy volunteer, for example. And this is what it looks like. This is what the actual chip looks like. You have colors, and typically red means that a gene is elevated in that person, and a green means that the level of the gene is lower than what it should be in that individual. The pioneering study is just illustrated here, was done by Dr. Mike Whitfield at Dartmouth. He took skin biopsies from patients with scleroderma, isolated the RNA, hybridized them through these little chips, compared them to healthy individuals, and measured all the genes that were present, all the 30 or 40,000 genes. Again, red means that a gene is elevated, and green means that it's down. The reds up here are the people with scleroderma, and these other branches are healthy individuals. And what's very obvious is that scleroderma genes are expressed very, very differently than in healthy individuals. What these genes are and what they do is a very complex story, and it requires very extensive analysis that's currently going on. Now, in a subsequent study, he made another very, very important observation. In this study, the Whitfield group took biopsies from a lot higher group of individuals, something like 20 patients and 20 controls, and again did this gene array. And what they found, and I think you can appreciate, is that there's tremendous heterogeneity. Different people with scleroderma actually look very different at the molecular level. They may feel the same, they may look the same to their doctor, but when you look at their molecular signature, they're very, very different. So this is really an extremely important observation, because it tells us that scleroderma actually has multiple subsets that we perhaps never really thought about, and by understanding what these subsets are, we might be able to develop more targeted and more specific treatments for these individuals. This is actually not a pipe dream, because, for example, in breast cancer, this is current clinical practice. If you have breast cancer, your doctor will take a biopsy, do these microarrays, identify specific patterns, and really base the treatment decision on what these molecular signatures show. And this is a very important study, for example, that showed that a set of 70 genes can accurately predict, if you have breast cancer, whether you need aggressive chemotherapy or whether a more conservative therapy will be fine. So this is actually in clinical practice, and these chips are commercially available and are increasingly used. So the hope is that if the data is validated, this kind of decision-making will enter clinical practice for diseases like scleroderma as well. So transcriptomics is one of the very hot areas, again, looking at all the RNAs that are being expressed. We have learned that we didn't know before, that even patients who look very similar clinically might have very different molecular signatures, meaning they probably need different kind of treatments. And the hope is that these transcriptomics will help us classify patients, guide therapy, identify which patients are appropriate for which drugs. In the future, perhaps clinical trials won't just lump all the patients together, but rather separate them out based on their molecular signature. And ultimately, this is moving towards this holy grail of personalized medicine, where we're not treating the disease, we're really treating the patient depending on exactly what their process is. Finally, a word about proteomics. This is simply looking at all the proteins that are present in the blood or the tissue. Obviously, the most easy way to do this is in the blood. It's actually very complex, because the blood has thousands and thousands of proteins, and many of them change during their evolution as well. But the idea of proteomics is to, instead of just fishing for a single protein in the laboratory, look at all the proteins and understand what their pattern is and what they are telling us about the disease. The first proteomic study in scleroderma was completed by Dr. Lafyatis and his group, and this is what the data look like. It's what's called a heat map, very, very complicated. Each column is an individual patient, and each row is an individual protein. And again, without going into detail, I think it's very apparent that there are very complex patterns that are almost certainly telling us important things about what's the underlying process in these people, and perhaps how we should be thinking about treating them. So I think many of us feel that this omics approach of the top-down, high-throughput approach, utilizing sophisticated technology, will really change the way we think about scleroderma, both in the research arena and as well as treatment. We can now measure DNA, RNA, protein. We can even start measuring what's called metabolomics, the metabolic profiles that change individual, and then put this all together in some predictive models. So it has a lot of utility. It fires up the research by identifying many novel targets that we can study that we perhaps never really thought about before. It helps us understand individual variability, allows us to risk stratify patients depending on how high a risk they are to develop complications like pulmonary fibrosis or pulmonary hypertension. It will probably make clinical trials much better because now we can hopefully identify patients who are more likely to respond to a drug and eliminate patients from a study who are very unlikely to respond to that drug. And ultimately, the goal is to really develop this into a personalized medicine approach where we can accurately match patients to the drugs that are most likely to work in those patients. And the challenge is really that we truly live in an era where there's a tidal wave of information, so generating the information and even analyzing it is not the limitation. The limitation is really understanding what all this information is telling us and how we can convert the information into useful knowledge to impact on better treatments.